Am I audible to everyone? Is my screen visible? Hello?
Yes, ma'am.
Yeah, okay. Okay, so let's start the live session for this week. Today I'll be taking questions regarding the week 6 video content of the course Tissue Engineering. So, in the week 6 videos, we would have seen about first cell adhesion, its key mediators, selectants, integrants, catherins, and so on. About cell migration, various factors influencing cell migration, and experimental approaches to study cell migration. Signaling and delivery of biomolecules like growth factors and mitogens, and delivery parameters and strategies that are involved. We would have seen about bioreactors, its challenges and prospects, and finally about challenges in engineering tissues, various types of tissues, that is skin tissue or cartilage, bone, and so on. So the first question is, which among the following cell to cell adhesion or cell to matrix adhesion molecules are calcium dependent? So select all that apply. The options are integrins, selectants, cadherins, immunoglobulin like receptors. So among these, few of the molecules are calcium dependent. So what could be the answer? Okay, so integrins, selectants, and cadherins, these are the class of molecules that come under calcium dependent adhesion molecules. Immunoglobulin like receptors are calcium independent adhesion molecules. So I have referred one paper to study further about this calcium dependent adhesion molecule, I mean the strategy that is followed. So in this article, what they have studied is they have taken cadherin. So we know there are two types of cadherins, E cadherins and N cadherins. So they have taken N cadherins and they have expressed that N cadherin protein on cell surfaces. So in two different cell surfaces, cell 1 and cell 2, they have expressed the N-cadherin protein and they have tagged it with different fluorescent molecules. So one is this, the C and V that we are able to see here. These are two different chromophores actually. V is for Venus and C is for another chromophore. So here what they have done is they have done the FRET analysis. So in FRET analysis, what usually is studied is that in this mechanism, energy is transferred between two different sensitive chromophores. So there will be a donor chromophore and an acceptor chromophore. So the donor chromophore, which is in the electronically excited state, will transfer its energy to the acceptor chromophore, and thereby we will be able to see the fluorescence intensity during this process. So here we are able to see that the cadherins, which are illuminated when fluorescence is the fluorescence intensity is seen between the cell to cell, showing the interactions, that is the localization of these membrane proteins, which is a proof for cell to cell interactions that are taking place. So these are all the fluorescent images, and these are compared with endogenous N-cadherin that is present in vivo. The second question, N-cadherins are seen in dash. So N-cadherins are seen in some particular cell types. So what are those cells? Hello? Okay, so N-cadherins are seen in nerve cells, heart cells, and lenses. So as I told, there are two types of cadherins, E-cadherin and N-cadherin. One main difference between these two are that the E-cadherin, the E-cadherin will bind to a shorter isoform. So there is another protein called catenin. So the E-cadherin will bind to a shorter isoform of this catenin protein, while in N-cadherin it will bind to a longer isoform of this catenin protein, that is the P120 catenin protein. Third question, periods when a cell is moving forward in a straight line without turning are called dash time. So this is a very straightforward question. It's taken from the cell migration video only. Can anyone try to answer?
From persistence time.
Yeah, it's a persistence time. So we would have seen in that video that we would have studied about the random walk of the cells. So initially the cell will move in a particular directed motion, and after that it will move in different random motions which is undirected. So the initial movement of this cell is, the time that it takes for this moment is the persistence time. So um, persistence time will differ based on normal cells and patient cells. So here is one example of that. This paper has studied about movement of cells from normal patient and schizophrenic patients. So the difference that they see is the persistence, that is the control cells have less persistence and patient cells have more persistence. The displacement, the period between turns, that is the path that they take, and the turn angles, their exploratory behavior, and the phenotype on the ECM substrate. So there is difference in all these parameters between a control cell and a patient cell. So we usually study this persistence time because when we are fabricating a tissue, we need to study cell migration, and cell migration in 2D will be different from cell migration in 3D. So to quantify this 3D cell 2D and 3D cell migration, we will study about this parameter called persistence time. Question number 4, dash coupling reaction engage, just a minute. Sorry, question number 4, dash coupling reactions engage the amino acids in the lysine residues to the N terminus of growth factors to cross-link with the scaffolds. So these are some examples of chemical coupling reactions. So which coupling reaction? Negishi coupling, Suzuki coupling, carbonamide coupling, free radical polymerization. So which coupling reaction will cross-link the amino acids in the lysine residue, the lysine residue amino acid, and the N terminus of growth factors. So N terminus of growth factors will usually have amine group, and that amine group will be cross-linked with carboxyl groups. So which chemical coupling reaction is involved here?
Is it carbonamide coupling?
Yeah, it's carbonamide coupling. So what basically happens here is that there will be many growth factors or ligands in the scaffolds that is used to attach the cells that are seated on the scaffolds. So in carbonamide coupling, so this carbonamide coupling is specifically between a carboxyl group and an amine group. So the answer is option C. What actually happens in this coupling, it is also called as EDC-NHS coupling protocol. It's very simple. So a carboxylic acid group that is present in all the amino acids, this will be cross-linked with a primary amine. So here we are able to see a carboxylic acid group and a primary amine group. So directly these two can't be cross-linked. When you just add an amino acid with carboxyl group, and when you add any other primary amine chemical, it won't be cross-linked, it will just remain as water. If you want it to cross-link and if you want it to polymerize and combine as one single polymer, you need to activate the carboxylic acid group. So for this we use a chemical called EDC. So EDC is a type of carbonamide only. When we use EDC, this EDC will form an unstable reactive acylurea ester with this carboxylic acid. So this unstable compound uh, paired with a primary amine to form a stable amide bond. So this is the chemical amide bond that is formed artificially, Or if it is reacted with water, hydrolysis reaction takes place and again it comes back to the normal carboxylic acid form. Or it can also be reacted with another chemical called sulfo NHS. So in EDC NHS, EDC you will use to activate the carboxylic acid, and you will use a second chemical called NHS. This NHS will replace the EDC. So, and it will form an amine reactive sulfo NHS ester molecule. So this will be an unstable reactive molecule, and this will be a stable molecule. Further on reacting with a primary amine, it will form an amine bond. So it is just a reaction for formation of an amide bond. But when you want to do it chemically, this particular coupling reaction will be used. So this EDC NHS coupling protocol is widely used in so many polymers. So basically when we see, for example, if we take hyaluronic acid is a natural polysaccharide. Hyaluronic acid has many COOH groups. And if you take gelatin, which is also a polymer, protein polymer, that has many amine groups. So if you want to form a hydrogel or a scaffold with these two polymers, we can use this EDC NHS chemistry. So once we use this, we'll be able to cross-link these two groups, and then once you freeze dry it, or lyophilize it, and then you cross-link it, you will be able to form a hydrogel or a scaffold. Any doubts? Moving on to the fifth question. Which among the following are in vitro pre-vascularization techniques? So the options are arteriovenous loop technique, cell seeding technique, generation of spheroids technique, or cell sheet technology. So which of these following options could be techniques for in vitro pre-vascularization, not in vivo? Okay.
Option B and C.
Yeah, option B and C, cell seeding and generation of spheroids are used for in vitro pre-vascularization. Along with it, option D, cell sheet technology. Sorry, I missed to mention the answer. So along with it, cell sheet technology is also used for in vitro pre-vascularization. So arteriovenous loop technique is used for in vivo vascularization. So here in cell seeding, generation of spheroids and all, what is usually being done is, so we'll have a scaffold. In scaffold fabrication techniques, we would have studied that scaffolds are formed with a lot of porosity percentage in them. So this porosity is for cell migration, cell proliferation, and vascularization process. So when we just seed these cells in a porous scaffold, automatically cells will start to multiply, proliferate, and it will start to migrate, and it will initiate the formation of, angiogenesis process will be evidently seen in the scaffold. So just with cell seeding we can see that. Using spheroids also, they are studying this angiogenesis process 3D in vitro. So one example for that is in this paper here. Here the paper title Xeno-free pre-vascularized spheroids for therapeutic applications. So this is from Scientific Reports. What they have done is, first, they have prepared spheroids. So the spheroids they have prepared with mesenchymal stem cells and endothelial cells. So for angiogenesis to take place, we need endothelial cells. So with these two, they have prepared spheroids, and in these spheroids, they have studied the 3D angiogenesis under fluorescent um, staining. So here we are able to see that there is a lot of sprouting from the spheroid. The sprouting is more compared to the EGM medium. So EGM is the basic gold standard medium that is used for cultivating endothelial cells. So apart from that, here in this paper they have used a xeno-free cell culture medium. So in the previous video that I have uploaded, I would have spoken about the xeno-free cell culture media and why it is important. So they have cultured the stem cells in this medium, and they have seen an increase in sprouting of from the spheroids that they have cultivated in day one itself. So this is one technique for studying 3D angiogenesis in vivo. In vitro, uh, if we want to study the angiogenesis process, one common assay that is being used in almost all the labs who are working with biomaterials is CAM assay. So I'm not sure if you would have heard of this assay. CAM assay is the Chig amniotic membrane assay. So we take a egg, and we remove the albumin that is present in the egg, and we incubate our material. So the biomaterial that we have fabricated, we incubate that material inside the egg, and we allow it incubated at 40 degrees Celsius, and we study the veins, the angiogenesis process that is occurring from the membrane. So the initiation of angiogenesis that is occurring from the biomaterial that we have incubated in it. So the main veins and the veins that are sprouting from that, and the other hair-like veins. All these veins are manually calculated. So if you upload this image in ImageJ software, we will be able to calculate the number of sproutings that is happening from the sample that we have incubated. So the vessel number, you can put it in a graph. So this also they have compared between the universal media and the xenobiotic free media. So um, these are like two methods that are used to study angiogenesis in vitro and in vivo. Any doubts? Okay, so the next question is, a team of researchers performed adhesion studies with the fibroblast cells. They observed that cells could adhere to the scaffold containing RGD ligand. The dissociation constant was found to be 1.7 into 10 power minus 6. What is the value of association constant? You can give the value in terms of 10 power 5. So, sorry, this is 10 power 5. So, in the videos, we would have seen that one of the major ligand that is extensively used to adhere fibroblast cells to the scaffold is RGD ligand. So, in this experiment that is being performed, they have given the dissociation constant, and they have asked to find the value of association constant that is prevailing there. So, there is one formula for this. Can anyone tell the formula? So this is the formula. Since both are inversely proportional, dissociation constant and association constant, when you divide the dissociation constant with 1. So when you divide this value, you will get the association constant. So can anyone tell the numerical value?
Ma'am, is it 5.8 into 10 to the power 5?
Yeah, yes, it's 5.88. So the answer is 5.88 into 10 to the power 5. Moving on to the next question. The picture denotes different cell junctions. Which of the following options denote correct ABC? So there are various cell junctions here. So which one of the following answer is correct? Adherence junction and gap junctions are already given in the picture. Apart from that, you have to identify the other 3 junctions.
Option B.
Sorry, option?
BB.
Yeah, yeah.
Desmosome, hemidesmosome and tight junction. Is it so?
Yeah, that's correct. So the A that is denoted here is desmosome, B is a hemidesmosome junction, and C is a tight junction. Yeah, so the answer is option B. The next question, which of the following events require the cell migration to happen? Wound healing, antigen presentation, regeneration of nervous system, destruction of infected cells. So these are few events that takes place in our body, and which among these events require cell migration to happen?
Option B and D.
Yeah, option B and D is correct. Antigen presentation and destruction of infected cells. So destruction of infected cells, usually our neutrophils and macrophages usually migrate to the place of infection and destruct the cells. For antigen presentation also cell migration is required, and along with it wound healing and regeneration of nervous systems also include extensive cell migration processes. Yeah, for these events to occur. So the option is all of the above. Question number 9. Which of the following mechanical signal would highly regulate endothelial cell differentiation? So we would have seen about different cell types of the body by now. Which one mechanical signal is highly important for regulation of endothelial cell differentiation?
Is it shear stress? Option C.
Yes, yes, it's shear stress. So there are so many factors that will guide differentiation of a stem cell to a functionalized cell. So for endothelial cells, one of the major factor that is being studied in the last few decades is that it can be the differentiation to endothelial cells can be achieved in a very high percentage with just varying the shear stress of the particular medium. So if you are preparing a tissue, um, if you mediate the tissue, the concentration of the polymers that you are using in preparing the material. Then the shear stress that is experienced by the cell growing in that particular material will vary, and based on that the differentiation, that is the end fate of that particular cell also will vary. So there has been so many studies regarding this. One major study that is being carried out is that with the polymer called PEG, polyethylene glycol. Polyethylene glycol will be um, methacrylated, so we call it as PEGDA, PEGDA. So in this PEGDA, different concentration of crosslinkers will be used. So when the crosslinker concentration varies, obviously the stiffness of the material, the mechanical property of the material will vary, and based on that, the differentiation ability of a stem cell to of its final functionalized cell also varies. So, with in tissue engineering, to study the cell differentiation, this particular phenomenon of shear stress is extensively being used. Question number 10. What are the necessary parameters to seed the cells in a bioreactor for tissue engineering applications? So, the answers are already there. Option A, media flow pattern and direction, cell concentration and seeding suspension, perfusion time, media flow rate. So, some of the important parameters to look into uh, before seeding the cells in a bioreactor for tissue engineering applications are these parameters. So can anyone give an example of bioreactors that are used for tissue engineering applications, or the products that we obtained by using bioreactors and stem cells and everything? Any one product or any one common application? Okay, so one a common example that is being practiced in almost all the biopharma industries nowadays is the cultivation of monoclonal antibodies, which uses Chinese hamster ovary cells. So these cells will be cultivated in large-scale bioreactors using serum-free media that we studied in the previous lecture, the xeno-free media. This media will be used to cultivate these cells for the production of monoclonal antibodies. So this is one example for using bioreactors. And in tissue engineering applications, so when you are fabricating any one particular tissue or even a small part of a tissue, you need constant perfusion. So perfusion of the media in order to avoid hypoxia conditions, a buildup of toxins, and so on. So you will require small scale bioreactors in order to cultivate a fully functional tissue in a bioreactor. So these are some parameters that need to be considered before seeding the cells on the material. Question number 11. You are interested in studying the cadherin, selectin, and integrin biology of 3D cell culture. Which hydrogel system would you prefer avoiding for your studies? So, option A is matrigel, option B gelatin, option C alginate, option D none of the above. Which hydrogel system will we want to avoid if we want to study cell-cell or cell-to ECM interaction? Can anyone answer? It's a
Is it alginate?
yeah, correct. It's alginate. So alginate is a polysaccharide that is usually avoided if you want to study cell-cell interaction, because one main reason is that alginate is very hydrophilic. So it's because of its highly hydrophilic nature, cells won't be able to attach on that surface. So we studied about contact angle in the previous lectures. Contact angle, yeah, biomaterials should be hydrophilic in nature, then only swelling will take place, cells will move inside, cells will attach, proliferate, migrate, and so on. And the biomolecules that you have loaded also will be diffused out of the cell if due to the hydrophilic nature and due to the diffusion of water in and out of the gel. But then if it is highly hydrophilic, that is if the contact angle is less than 15% or 10, I mean 15 degree or 10 degree, then that will also not be helpful for cell attachment and migration. So since alginate has that high hydrophilicity, and it also does not have any cell interactive domains also. So due to these reasons, alginate is usually avoided when you want to study cell ECM attachment and all. Unlike gelatin and matrigel. So matrigel, we all know it's a material that is prepared from ECM, mouse ECM, so um, which has high cell adhesion. And gelatin, it's a protein, it has various amino acids, RGD domains, and it is used to study cell adhesion. So since alginate is not used to study cell adhesion only, we are using this alginate beads. We would have studied about sodium alginate cross-linked with calcium chloride to form calcium alginate, and these beads are mainly used for cell immobilization or enzyme immobilization. So they are used for immobilization because they can be entrapped inside that particular compound without reacting with that compound. So it will just be immobilized inside. It won't attach to the particular um, alginate bead. So that is why alginate is used in these applications and not in studying cell adhesion. So the answer is option C. Question number 12. In order to study cell migration properties, which among the following assays can be performed? So the options are scratch assay, Boyden chamber, micropattern based assay, fluorescent labeling followed by live cell imaging. So which among these assays should be performed if you want to study cell migration?
Ma'am, is it scratch assay?
Yes, scratch assay is the basic assay that we perform to study cell migration. Apart from that, the other three options, option B, C, and D, are also used to study cell migration. So let's look about let's look a little about these assays. Mm, So first is the scratch assay or the scratch wound assay we call it. So what we do is we will take the well plates, that is the 6 well plate that we have for tissue culture. We will seed the cells on the well plates, and once the cells are confluent, that is once they adhere to the well plate and form an even mat, we um, perform an artificial scratch on the surface of the well plate using a pipette tip. So we use a 20 microliter pipette or a 100 microliter pipette tip, and we we artificially prepare a scratch on that surface, and we study for the consecutive next 2-3 days how the cells migrate and how the scratch area is closed. So this is the scratch wound assay that we use for to study cell migration. The next assay that we saw is the Boyden chamber. So here how we study cell migration is that, so first we take a well plate. So in the well plate, we will place a semi-permeable membrane, and on the membrane we will feed the cells. So if these cells are able to cross the membrane, and if they are able to, so if cell invasion happens, then that means that cells are migrating. So after some time, cells from this membrane migrates to the media or the DMEM, FBS media, whatever media is there underneath, and then we will remove the semi-permeable membrane that is placed on top, and we will quantify the number of cells that are present in the media using cell lysis buffer. So through this we will be able to see the potential of the cells to mediate through a semi-permeable membrane. The next study is micropatterning study. So this is one interesting study that is being used to study cell migration after the invention of 3D printing. So in this particular paper, what they are doing is, first they are preparing gelma. Gelma is gelatin methacrylate. So methacrylated form of gelatin is gelma. They are preparing the gelma polymer first, and they are 3D printing the gelma polymer in parallel lines on top of PNIPM polymer. So that is a thermosensitive polymer, synthetic polymer. So it is thermosensitive in the sense it will be solid at body temperature, and it will be in a liquid at fridge, that is 4-degree temperature. So in this synthetic polymer slab, what they are doing is they are using a 3D printer to print parallel lines using the gelma polymer. So as we all know, gelatin has a lot of cell adhesion moieties, and once the cells are seeded on this plate, this particular plate, cells will be seeded all over the plate. So to eliminate the cells that are seeded in between the parallel lines, what they are doing is they are decreasing the temperature. So when the temperature is decreased, the cells attached on so cells usually will attach and grow only in 37 degrees Celsius. When the temperature is decreased, the cells will be viable, but it will be detached from the surface. So once it is detached from the surface, it will be washed away, and we will be able to see that cells are attached only in the gelatin. So even at that temperature, it is attached to gelatin because of the ligands, the domains that is present in gelatin. So after that, they are incubating this particular material in 37 degrees Celsius, and they are studying the direction of cell migration, the ability of cell migration and the direction of cell migration. And they are able to see here in this study they have used myoblast cells, that is the muscle fiber cells. So they are able to see that the myoblasts are migrating in perpendicular direction to the gelatin in the in between spaces, and they are migrating in parallel direction on the gelatin. So um, even the direction of migration of cell is it is varying based on the polymers that we are using. So this in this one particular study they have, with this study they have quantified, I mean they have qualitatively analyzed how cell migration takes place. Any doubts?
No, no.
Okay. So question number 13. Your project involves developing an organ culture bioreactor system. What are the challenges you anticipate? If you want to culture a whole organ, maybe for example a kidney. So what are the challenges in a bioreactor? What are the challenges you would anticipate? The major challenges.
Ma'am, first three options.
Yeah, that's correct. So first option, option A, nutrient delivery. As we all know, it's unlike 2D cell culture. 3D cell culture will face a lot of hypoxia conditions inside the tissues. So nutrient delivery and oxygen delivery throughout the organ should be consistently maintained in a bioreactor. The cost of culturing an organ. So as I told, perfusion bioreactors will mostly be used, and for that we need continuous supply of media. And media, a normal DMEM media which has FBS uh, or other serum components will be very costly, even for a 2D cell culture. So if we want to do a 3D cell culture, then we need media in large quantities. So cost is one another challenge, and maintaining aseptic culture conditions. So basically, so doubling time of a fibroblast cell, a normal fibroblast cell, is around somewhere around 12 to 18 hours. So just for doubling we need a lot that much time. So if you want to fill the biomaterial 3D construct that you have prepared with cells, it will take a lot of time, and maintaining the aseptic culture condition throughout that time will be a challenge. So these are the options. Wide availability of cell source won't be a challenge, because the cells that we are using, they are not primary cells, so we are not isolating the cells and we are using, we already have cells that are programmed genetically to multiply indefinitely. So when we are having cells like that, we needn't worry about cell sources. We can use the same cells for numerous number of passages, and we can collect the cells, all the cells, and we can seed them on the material. So the answer is option A, B, and C. Yeah, so these are all the questions for today. If you have any doubts, you can ask.
Ma'am, I have a question.
Yeah, yeah.
Ma'am, regarding our examination, like it's coming like 23rd, it seems. Ma'am, the
Yes.
question pattern, how it will be set? Like, can you give us an idea about it?
The questions will be like the questions that you're getting in your assignments only. Similar, maybe MCQs and fill in the blanks, match the followings, and so on. Nothing apart from that.
Oh, okay, ma'am. No, because it was written like it was pen and paper test. That's why.
Okay, so I think maybe this time they are trying to have a pen and paper test. Like the exams are taken care of the other teaching assistants. So maybe I'll talk with them and I'll let you know the
Okay, ma'am.
pattern and the.
Okay, ma'am. Also, ma'am, like
Yeah.
it was given in the announcement today. Like we will also receive some like essay type questions. So, ma'am, can you in the next week's class, like can you please bring some questions like the essay type questions so that we can get an idea like what would be the way in which the essay type questions would be set.
Okay, yeah, sure. Okay, that can be done. So, as I told, I'll talk with the other teaching assistants also who are preparing your questions and assignment questions and exam paper. I will have a word with them, and yeah, maybe next in the next live session, I can give examples of those questions.
Okay,
That won't
ma'am.
be an
Thank
issue.
you.
Um, Yeah, so I will change the meeting settings from the like for the next two meetings, so that you can enter even if I'm not there in the meeting. So last two meetings I was not able to conduct as I was out of station. So I will change the meeting settings accordingly so that you can enter or you can have a discussion. I will arrange the other TAs to be present if I'm not there. So there are only remaining two more live classes, and I'll be there for those classes. In case of any emergency, I will change the meeting settings and the meeting will go on. So I have uploaded the recorded videos for week 4 and week 5 video contents. You can have a look at them and you can post your questions in the discussion forum if you have any doubts.
Okay, ma'am.
Yeah, so we can end the session if there are no more doubts. Thanks a lot, everyone.
Okay, ma'am.
Thank you,
Thank
ma'am.
you.